All right. Yeah. I'm Dr. Mr. Ferguson, one of the researchers here at IBIS, and I'm here to tell you a bit about some of the research that we do here that you might not think about. While our scientific findings are usually the most eye-catching, there's a lot of work that goes on behind the scenes to develop and improve the mathematical methods that we use to get these results from the data that we collect. And just like improving the scanners we use can result in higher quality brain scans, improving the mathematical methods we use can help us see things in the data that we couldn't before. Over the past few years, IBIS has been spearheading development of a new method for finding connections between brain activity and behavior, called functional connectivity enrichment analysis, and we've just recently published a paper that statistically validates this method. Functional connectivity enrichment analysis has already enabled us to find results that we couldn't have found with previous methods, and as our newer data sets roll in, we're excited to see what else it allows us to find. So for this kind of methodological research, when we're developing and refining new analysis methods, what kind of problems are we trying to address, and how do we go about addressing them? While every particular sub-branch of science has its own set of analytical tools that come with their own set of statistical concerns, one overarching umbrella that many branches are grappling with these days are the problems of big data. Traditionally, scientific research would look at the relationship between a couple of variables at a time. For example, we might compare the height and weight of a bunch of people to try to find a simple relationship between the two. And traditional statistics was shaped by these kinds of analyses. However, with technologic improvements and ever-increasing scientific knowledge, the amount of data that we're able to look at grows larger and larger. For example, when we here at IBIS collect brain scans, we can see the functional activity at hundreds of thousands of 3D pixels in those images. And trying to use the same statistical approaches to analyze the relationship between the behavioral variables that we're interested in and all of those points of functional brain activity can lead to problems. Two of the main problems that come up when testing that much data at once are the problems of false positives and false negatives. False positive is when an analysis method tells us that there is a relationship between two variables, but in truth there is no such relationship, and it only appeared so by chance. To address this, we would normally just use stricter thresholds for saying that a relationship is significant. However, this then results in false negatives, when our analysis method misses a relationship that is actually there. Using statistical methods that are designed for smaller sets of data on the type of big data that we collect leaves us constantly trying to balance these false positives and false negatives. So the idea behind enrichment analysis, functional connectivity enrichment analysis, is to find a way to group all that data in advance, using what we know about the structure of brain activity to pool various statistical tests together, and thus reduce the number of tests that we're doing. For example, we know that the brain activity of all of these various regions in the back of the brain will actually be quite synchronized, as these regions are all working together to analyze what our eyes see. In fact, we've seen that functional brain activity synchronizes into rather consistent sets of these functional networks, allowing us to group all the regions across the brain into a dozen or so functional networks. So rather than testing each small region independently for its relationship with whatever we're studying, enrichment analysis takes all those individual tests and combines them across functional networks into single tests. By combining things in this way, we hope to be able to reduce false negatives and false positives at the same time. Now, while the method was designed to try to solve these problems, we don't actually know if it does until we statistically validate it. That's what the paper we recently put out does. By generating artificial brain and behavior data with a relationship that we're able to control, and then using both enrichment analysis as well as previous methods, we're able to directly measure the comparative rates of false positives and false negatives, and we're able to show that enrichment analysis improves on things in the ways that it was designed to. This sort of validation proves to both us, as well as to the wider research community, that this is a reliable method whose results we can trust. Since we've started working on enrichment analysis, we found several brain behavior relationships with it. In our infant group, we found brain relationships with restricted and repetitive behaviors, motor skills and walking, and joint attention, among others. And with our school-age kiddos, we found brain activity associations with social behavior. We're also using enrichment analysis in creative ways to put our brain data in conversation with other types of data. For example, we're using it to look at connections between brain activity and genetics, trying to explore the developmental pathways that lead from genetic predisposition to brain differences to the behaviors that we commonly associate with ASD. All in all, enrichment analysis is an exciting new method for finding associations with brain activity. We keep working on improving the methods that we use so that we can make the most of the great data that our research and your families have worked so hard to collect.